Assalamualaikum warahmatullahi wabarakatuh. My name is Isha Humaira binti Ma'az Jazili and my student ID is 2025890432. Today I will talk about plant propagation. To begin with, what is plant propagation? Plant propagation is the process of increasing the number of plants of a particular species or cultivars. Plant propagation is done through two main methods, which are sexual and asexual reproduction. Plant propagation is important because we can gain many benefits from it. One of it is it can cause efficiency, which reduces the need to buy new plants or seeds. Plant propagation also can produce genetically identical plants, which then ensuring consistency in desirable traits. It also improves hybrids and cultivars with better characteristics, including disease resistance. Plant propagation also supports plant conservation, which then allows large-scale and faster plant production. Next, I will briefly talk about sexual reproduction, which is one of the methods done through plant propagation. Sexual reproduction involves the fusion of male pollen and female egg gametes inside the flower. Sexual reproduction has its own process. It begins with pollination, where pollen is transferred from the anther to the stigma. Pollination can be self-pollination or cross-pollination. Pollination is assisted by pollinators such as insects, wine, water, and birds, which then can form fertilization. Fertilization occurs when the sperm fuses with the egg inside the ovum. The fertilized egg will then form a zygote, which develops into an embryo within a seed, and then the ovum becomes the seed, while the ovary develops into a fruit. This can produce genetic variation, which is important for plant adaptation and survival. Next is about asexual reproduction, which is the second method of plant propagation. Asexual reproduction produces plants genetically identical to the parent with no seeds or gametes. Its natural methods are tubers, bulbs, rhizomes, runners, budding, and fermentation, while its artificial methods are cutting, which is rooting stems, leaf or tips, grafting, that is joining scion to rootstock for new varieties and disease resistant, and lastly layering, which is rooting a stem while still attached to the parent plant. Next is about tissue culture. Tissue culture is a biological technique used to grow plant cells, tissues, or organs in a sterile and it has a controlled environment outside the parent plant. Tissue culture uses nutrient-rich media in gel or liquid form. Tissue culture also has its own advantages. One of it is tissue culture can have rapid multiplication of plants. Then it can produce genetically identical and disease-free plants. And lastly, it allows research on plant cells and tissue. This means that tissue culture is an important tool in plant propagation, biotechnology, and scientific research. Next, I will talk about the special structure of a plant in plant propagation. To begin with, we'll start with stolons, that is also called as runner. Stolons is a horizontal stems growing above ground, and its roots and shoots form at nodes, which then can form in new plants, for example strawberries. Next is rhizomes. Rhizome is a horizontal underground stem, and it has sections with nodes, produces roots and shoots, for example ginger, iris, and ferns. Next is tubers. Tubers is a modified stems or roots, and it has a cut section with eyes developed into new plants. For example, potatoes, dahlia, and cassava. Next is bulb. Bulbs propagate either offsets or artificial method. which is scaling, chipping, and scoring. Bulbs can be produced genetically identical plants. For example, onions, tulips, and lilies. Comb is a special structure that can produce smaller combs or comals, or can be divided, and each piece with a bud grows into a new plant. For example, gladiolus and alocasia. And lastly, rootstock division. Rootstock division is a structure that divides specialized roots such as tuberous, swollen, and rhizomatous. And each section with a bud can form a new plant. For example, dahlia, sweet potato, and ginger. Nowadays, people use asexual reproduction as the main method in plant propagation. This is because it has many advantages. One of it is fast and energy efficient, which means that it can reproduce quickly without a mate. Asexual reproduction also preserves beneficial traits. For example, like offspring, they are genetically identical to the parent. It also has a minimal resources required for structures like tubers, bulbs, and rhizome that can help survival of a plant. Asexual reproduction also has a rapid recovery. In this way, plants can regrow quickly after the damage. It also has an ease of propagation, which means that it can be useful for gardeners and farmers especially. Lastly, asexual reproduction has uniformity, which means that it can ensure consistent quality in agriculture, aiding harvesting and control. However, it also has its own disadvantages. One of it is asexual reproduction has no genetic variation, which means that all offsprings are clones of the parent. It also has vulnerability to disease or pests, which in this way that entire population can be affected simultaneously because of it. Asexual reproduction also has limited adaptability. This way, plants cannot adjust to new or changing environments. Next, asexual reproduction also has transmission of defects, which means that disease or harmful mutation can pass directly to the offsprings. Asexual reproduction also is overcrowding because of its rapid reproduction that can lead to competition for resources. And lastly, it can reduce vigor over time. In this way, it means bad for it because it can be repeated cloning that may shorten lifespan and weaken the plants. That's all for me. Thank you for watching.